Questions?
So to what extent does stimulation of non-neuronal elements, many of which have membrane potentials, play a role in any of the effects that you see?
Yeah, that's a great question. So we don't really know what is the neural substrate of the effects that we see to begin with. In the case of the motor evoked potential, one can hand wave around and say, well, somehow or another pyramidal cells are activated, and that certainly is the case, but we know that even there, a lot of the effect is through interneurons, and we can show that even in the animal models with recording. I think there is pretty good evidence that in addition to that, there is effects on glial cells, and that some of the effects of the modulatory lasting effects that we see are probably mediated by those glial effects rather than by the direct neuronal effects. So that has been looked at. The question was, what about vasculature? And that has been looked at least to the extent that it can be with imaging in humans, and there is no change in perfusion, no change in regional cerebral blood flow, no breakdown of the blood-brain barriers, as has been shown with ultrasound, and no change in FDG that is not matched to an activity change. So a primary vascular effect doesn't seem to be the driver, but it can certainly increase the effect because of a secondary effect.
Hi. My name is Hanne Stensola, and I'm a postdoctoral fellow here at the Champalimau. I have a question regarding deep brain stimulation and Alzheimer's. We know from rodent work that the medial septum is essential for the expression of grid patterns in entorhinal cortex, and also as long as the environment is novel and large enough for place field expression. Is it a target for human deep brain stimulation?
The entire brain is open for business in terms of potential targets. With respect to the medial septum specifically, though, indeed, because the fornix is the bidirectional highway, the axons from the medial septum that are cholinergic, glutamatergic, and GABAergic are stimulated as they project to the hippocampus. And so part of the effects we see might indeed be related to effects on the medial septum hippocampal system. So it is indeed a target. We are stimulating huge assemblies of axons with multiple, N-terminal termini, and so we have not been able to dissect which of those elements and which of those pathways is important and which one should be avoided. This is going to require techniques other than stimulation, which just affects everything in the vicinity, neurons and axons. So to answer more specific questions as to what's the role of a specific nucleus or specific structure, we're going to have to go to methods that are more discrete, for example, optogenetics, to selectively activate that particular pathway.
Hi, I'm John Ioannis Sotiropoulos from ICVS Institute in Medical School of Minho here in Portugal. So based on the fact that what the main problem of an AD brain is basically the loss of synaptic plasticity, the loss of the ability to form new recent memories, not so much with the retrieval of back old ones that are already formulated synapses that's still okay. So what is the evidence you have? Do you have any effects on LTP, LTD, or even in the circuit and synchronization between areas, which is important for retrieval of memories? And how would you see your deep brain stimulation or even the other mechanisms that you suggest today in a way that you could block the neuronally activated, at least driven, propagation of tau pathology, for example, or from entorhinal to the other. So can we use it prophylactically in a way? Yeah. So when you see tangles, when you see the damage coming, can you do it in entorhinal? Can you block somewhere through the neuronal activity before it is too late? So it was 2 questions.
So what we're seeing is that deep brain stimulation makes more neurons, makes more branching of neurons, and makes more synapses. We also know that stimulating these circuits improves memory function in the animals. So their memory is enhanced. So our hypothesis is that, indeed, we are rebuilding circuits and repopulating synapses that are lost. So we think that one possible mechanism through which this may work is indeed to restore the number of synapses and synaptic functions. As you know, the cognitive deficits in Alzheimer's closely are related to synaptic numbers, and we think that we might be able to increase the number of synapses, and if those synapses are working, there's a chance that we might be able to turn back some of the deficits seen in Alzheimer's.
If I may just offer also from the transcranial magnetic simulation, I tried to show in humans, we know, in fact, that there are changes of the LTP-like measures that we can demonstrate and that I think Albino will talk about this afternoon in more detail. But after this intervention that includes brain simulation and where the brain simulation is a critical juice component of the intervention. In the animal models studies that Alex Rothenberg has been doing, the same is true. And in fact, he can show that there is an improvement in synaptic plasticity mechanisms and an increase in synapses, just like with deep brain stimulation. I think the question becomes, is that really driving the toxicity? And if you reverse it, would it change anything? If the answer is yes, like you seem to imply, then yes, one could potentially explore it as an early preventative sort of intervention. But I think that is not clear at all, as far as I know. At the very least, it may be, to some degree, a disease-modifying effect. Some of the data seem to suggest that, but I think it's super preliminary. It hasn't really been looked at that way.
Yes, Isabel Fariñas from University of Valencia. Thanks for this terrific session. And I would like to go back to DBS and the effects on neurogenesis. I was wondering whether about the potential long-term effects of that activation, because these neural stem cells that sustain neurogenesis may become prematurely exhausted in a way. Have you done in the experimental animals long-term analysis of that neurogenesis?
Our analysis is only in the short term, in the order of 3 months. So we know that there's a net increase in the number of cells. There's, for example, we do caspase staining and it's relatively low. So there's a net increase in the number of cells. The cells are incorporated in the circuit. They have normal morphology. They can be activated with memory tasks. We see CFOS labeling. So they seem to be functioning, and they seem to be imparting also improved memory in the animals. If we block neurogenesis by giving temozolomide, a mitotic blocker, then those effects are lost. So we think that there are some functional consequences to making new neurons. The issue is, do patients with Alzheimer's make more neurons? So we've not been able to answer that. We can certainly answer it in rodents and rodents with transgenic, but as mammals age, the propensity for neurogenesis drops. And so we're dealing with humans that are 60, 70, 80, 90 years old. We do not know to what extent they have preserved ability to make new neurons in their brain. So in theory, in the animals, it works very nicely. Whether this is true in humans, in elderly humans, we don't know.
In fact, you don't seem to see a rise in astrocytes. And we tend to think that astrocytes are, some neural stem cells seem to be associated with astrocytes, might give us some hesitation in going into neurogenesis in adults. I had a question. So you showed the effects of TMS with non-pharmacological cognitive stimulation, and then you show them with different pharmacological agents. Actually, it's 2 questions. One is, I know these are two studies. Could you discuss the difference in the ADAS-Cog in both of them? Since it's a 4-point difference in the latter, and I believe ADAS-Cog has got about 150 points. So those 4 points, how much do they matter for the patient when we think about bringing this into the clinic? And then you see so the interplay of TMS with different pharmacological agents, some of which are known to have similar effects, seems to be markedly different, at least. I don't know how significant it is. I know those are preliminary data. So would it mean that TMS would work better with some agents working in some pathways and not with others? And which would these be?
Yeah, let me just first make sure that I clarify what the slide shows, because I apologize. I think it led to a confusion. So I was showing the results of this TMS with cognitive training intervention as compared to the results of trials of the medications. So it's not, so the other curves were simply to illustrate what do you get in the same patients, the same patient population, with medications, which is about a 2-point ADAS-Cog improvement that is short-lived. What the results of the TMS plus cognitive training show in this pivotal study is that the effect is larger than the medication trials and that it lasts longer, which is great. Now, admittedly, as I was pointing out, those were patients on medications. And so the point that you're making is well taken. It could be that it is precisely because they were on medication that there is an interaction. And in fact, Katie Brem has looked at this a little bit. There is and published a small sort of a study showing that if patients are on memantine or on acetylcholinesterase inhibitors, the modulatory effects of TMS actually are different. And so I think that very question is an important one, and one that stands to be explored still is, are the effects of even this trial differential depending on what medications the patients have been on, and that could very well be.
I don't know if this is Albino's territory, but does this 4-point difference make a significant difference to patients or not? I mean, it's obviously very promising and worth exploring.
I'm happy to answer. So the other thing that we got as outcome, in addition to the cognitive training, is actually an activity of daily living question, because it is well known that the ADAS-Cog is not a suitable scale to really assess to what degree a given change is meaningful. Again, to put in context, on the ADAS-Cog, you're correct in terms of the size of the scale, but 2 or 3 points in a change because of medication actually translates into a change in the clinical activity or daily activity scale that would be meaningful, and the same was true here. So if you look at the CGI and similar scales of activities of daily living, severity of disease impairment and disability because of it, the patients did show an improvement that was in itself significant, independent of the ADAS-Cog.
I was hoping to have a question also, but the question is, it seems like this option for non-invasive brain stimulation is closer or potentially closer to the possibility of entering regular use, or at least to being accessible. Where do you think it fits in the treatment circuit for these patients? Where will it go? Which patients?
So it hasn't been studied in MCI, and I think it's a worthwhile study to do, because it may well be that the effect is even greater earlier. And as I was mentioning sort of in passing, when you look at moderate versus mild, the effect is significantly greater in the mild than in the moderate patients. So I think that's probably the target patient population. Whether or not it is in addition to or before or after medications, I think is a question to still be explored. But I think it's early in the course of the disease when activities of daily living start being impaired, and that probably the target is. In terms of clinical adoption, I think there are still many questions to be had. This is a short effect study still, and how long it lasts, or whether it even sustains a benefit or even reverses, whether long term it changes. I think these are important questions that we don't know yet. And patients here were treated in one single course, and they showed a benefit still at 3 and at 6 months. But for the most part, we've actually followed the patients that we entered in our trials, and at the end of 1 year, they don't really seem to show any benefit, raising the possibility of maintenance or of more invasive interventions. So I think those lasting effects need to be at least explored before one adopts them widely into the clinical practice, I think.
One final question. Thank you very much for the session and with regard to expectation. Brian Strange from the Fundación 100. A question for Andrés. The vascular effects of the stimulation are really quite striking. So I just wonder whether you might have perfusion MRI scanning on your patients, or arteriospin labeling, or what have you, to see if this vascular changes can translate into some anatomically quantifiable effects.
So, yeah, you're quite right. So we think that making new blood vessels is an intrinsic feature of deep brain stimulation, and we think that it's mediated through the stimulation driving the expression of vascular endothelial growth factor. So you stimulate, you make vascular endothelial growth factor, and that makes blood vessels grow. This, in turn, makes the brain expand. And in fact, we think the majority of the growth that we're seeing is related to the blood vessels. We expect that to have some functional consequences on brain function as well. But as of yet, we have not examined that. So we hope to be able to try to figure out, what are the consequences of making more blood vessels in your brain. Could it cause hemorrhages? Could it, could it help with, the clearance of toxic things? So, the whole universe is open as to what are the functional consequences of making more blood vessels in these areas of the brain.
Thank you very much for the speakers for an outstanding session. And we will be back at 2:30.